Welcome, welcome, ladies and gentlemen. We're talking about movement through the membrane today. So, key questions: what determines the direction in which passive transport occurs? Why is osmosis important? And how do substances move against their concentration gradient? Two types of transport. We've got passive transport and active transport. Let's get some definitions down first. We have equilibrium, when the space is filled evenly, or it is the same on both sides. It's going to be equal. Now, a concentration gradient is the difference in concentration of a substance across a distance. So when there's more on one side than the other. Now, when talking about passive transport, there is no energy required. It's passive. It's laid back. It's kind of chill, and it just sort of occurs. No energy is put into it. It just happens. Passive. Now, there's going to be lots of different types of passive. So let's talk about diffusion. Diffusion is the movement of molecules from a high concentration to a low concentration. Again, a high concentration to a low concentration. Molecules in a solution are constantly moving. And when they move from a high to a low, that's diffusion. No energy is required. This is simple diffusion. Moving straight across the membrane is simple diffusion. Again, moving from a high to a low, simple diffusion. Facilitated diffusion requires a transport protein to help move the substances through the membrane. Again, passive diffusion is facilitated diffusion is passive transport, no energy required. Transport proteins, such as the channel protein, have this channel here that allow things to move backwards and forwards, in and out, no energy required, from a high concentration to a low concentration. And carrier proteins, high concentration, it comes in and it lets it out the other side to a lower concentration, facilitated diffusion. One more time, movement through the membrane, simple diffusion. Movement through a carrier protein, such as a channel protein, is again simple diffusion because it's moving in and out. Then we have facilitated diffusion, which is a carrier protein, which takes it in and then it moves it along to the other side. Osmosis is the diffusion of water. So again, this is going to be passive because it is a type of diffusion, specifically talking about water moving across a semi-selectively permeable membrane. Selectively permeable means only certain things can go in and out. And again, it's movement of water from a high concentration to a low concentration through a protein channel. Water is constantly moving back and forth through a selectively permeable membrane. But again, when it's moving from a high concentration to a low concentration, this is going to be osmosis. Isotonic solutions are when the cell, the concentration of the water on the inside of the cell, concentration of the water on the outside of the cell are equal. Also, the concentration of any of the dissolved substances inside the cell is equal to the dissolved substances on the outside of the cell. So there's just as much water on the inside as there is on the outside, and there's just as much salt as there is on the inside as there is on the outside. Isotonic means equal. Types of solution, we are always going to look at the water. The water, how much water is inside or outside of the cell. Using your root words, iso means equal, hypo means below, hyper means above. Let's take a look at the hypotonic and hypertonic solutions. A hypotonic solution is when there's more water on the outside of the cell than there is on the inside of the cell. So if there's more on the outside and it wants to be equal, then we have water moving through, causing the cell to get larger. So hypo with the O, water comes in and the cell will grow. Again, hypo with the O, the cell will grow, and if too much water comes in, it can cause the cell to burst. In plants, they have a cell wall, so they're not going to always burst, but the pressure on the inside of a plant cell is called turgor pressure. This is the force exerted outwards on the cell wall by the water contained within the cell. This gives it the plant's rigidity or structure and strength. So again, hypotonic Hypo with the O, the cell will grow. So that means water is moving into the cell. Hyper is the opposite. Hypertonic solutions, we have water outside the cell is lower than inside the cell. So to make it equal, water will move out of the cell. Water will flow out, and the cell will tend to shrivel up or get smaller. In a plant cell, when it loses its water from the central vacuole, the plasma membrane shrinks away from the cell wall and the plant wilts or loses its turgor pressure. Again, when there's too much on the inside and not as much on the outside, it'll have water move from the inside to the outside, and it will shrink. The cell will shrink. So, in this example, we have water moving into the cell. So that's going to be a hypotonic solution. Active transport. Active requires energy. So it's movements of materials through a membrane against the concentration gradient. So that's going from low to high, not high to low. This requires energy. The protein carriers actually need the energy. So how does this work? Carrier proteins bind to a particle that is transported. Chemical energy, ATP, allows the cell to change its shape of the protein, and the particle moves into the area of a higher concentration. Let's look at this in a picture. So, there's not as many ions as they're out here. So this is a higher concentration. The ion moves in, and the carrier protein changes position since it's inside, and energy allows the carrier protein to snap and open to the other side, releasing the ion to a higher concentration. After the ion leaves, the carrier protein reverts back to its original position. Again, active needs energy. Endocytosis. Endo coming in, cyto meaning cell. So this is the process by which a cell surrounds and takes in material from the environment in a vesicle. The membrane formed around the material is a vesicle. Phagocytosis. Phago meaning eating, cyto meaning cell. So it's cell eating. Cytoplasm surrounds a particle and packages it within a food vacuole or vesicle and engulfs it by the cell. So it's engulfed by the cell. Phagocytosis is typically solid substances. Pinocytosis, where did my mouse go there? Pinocytosis is going to be liquid substances processed by taking in liquids from the surrounding environment. So again, material moves in, the plasma membrane bulges, it fuses to create a vesicle with the material inside of it, endocytosis. Exocytosis is the exact opposite. It is the expulsion or secretion of the material from the cell, such as waste or hormones. It's the exact opposite. So we have something we need to get rid of. The vesicle comes up, fuses with the cell membrane, and then releases the substance. So let's take a look at this. And releases the substance. This is exocytosis. Let's take another look. Fuses, releases the substance. Exocytosis. That's all I got for you today. Bye.